I am Mikhaela Neequaye. I came to Gatsby Plant Science Summer School in 2013, so that's me exactly where you were 12 years ago. And that is me technically taken 2 years ago at the field site that I left on Saturday. So doing the plant science research that I'm currently actively doing and was in fact doing last week in the Italian Alps. So I'm going to talk you through a little bit of that. I thought I'd do a bit of career stuff because I felt like this was the moment, because my journey's been a bit non-canonical, but it started that way. So I thought I was going to I did a bachelor's in genetics. I actually thought I was going to cure cancer. I think everyone did. And I stayed very committed to genetics, not thinking so much about the models I was going to be working with. And then I came to the Gatsby Plant Science Summer School, and I thought, oh, people actually do genetics in plants. And so that's what I decided to do my PhD in. What I did before that is between my second and final year, I got a funded studentship from the Genetics Society, so I actually got to do research in a lab, fully funded, which was incredible. And that was where I really got the bug and thought, oh, I can actually do this. I did my PhD at the Quadram Institute in the Norwich Research Park on broccoli. I'm really good at broccoli. And then COVID happened, and my visa to go to the US to do a postdoc was suspended. It was the first time Donald Trump was the president. And I ended up working for a really cool NGO called Two Blades. I was in crops, and I thought, oh, I don't think I want to stay in crops. And I became a postdoctoral research associate in the Byers Lab, which is where I am at now. So what does the Byers Lab do? I technically am a chemical ecologist. I like to joke and say that I study, the chemistry of chemistry. And that is to say, I think about chemical attractants, specifically all of the biochemical ways that plants try and make themselves attractive in order to mediate sexual reproduction through a third party, and that is pollinators. Now, I believe that you heard from Edwige today, so you know a fair bit about some of the ways that plants create structures that are beautiful and visibly enticing, right? And they do that with chemicals as well. They make gorgeous colors, red if you want to bring a hummingbird along, yellow if you want to attract a bee. But when you're flying around a meadow, a bumblebee, a wasp, a solitary bee, a fly, there needs to be that initial go over there, right? So that's what I focus on. Our lab works on scent predominantly. And the first thing we have to think about when you study floral scent and this chemical attraction is, how do you capture a smell, right? How do you know what something actually smells like? Because it's so subjective. So again, this is the field site we were at last week. We trap smells. This is one of our systems. This is the orchid. This is the Gymnodenia nigritella. It's also known as the vanilla orchid. You walk into the fields and it smells like chocolate and vanilla. And it's because of these orchids. So what we do is, it's very technical. We wrap them in a chicken roasting bag. And then we use a pump to suck the air out over 24 hours. And we trap that in a solvent. And then we do some really cool, but very basic chromatography, right? I think you guys have ever done the experiment where you put an M&M on a tissue paper and you see the way the colors separate. Same thing. It's exactly the same premise, except for we use a gas, usually just an inert, mobile gas. I think it's usually helium. We press our sample through the GC system. It moves around according to the size and the dynamics of the chemical that we're looking at. And it comes out into an MS detector, GCMS. It is the lifeblood of what we do. I'm happy to talk more about it. And what it produces is this. This is a chromatogram. You think it's really boring. It's not. I always delight in seeing them. And even though I've been looking at them for 5 years, they're still fantastic. So this is the other system I work on. I'll talk to you about it. It's Mimulus, the monkeyflower. So this is Mimulus lewisii. This is the model for what a bee likes. And I think some of you might end up learning about it. I know I learned about monkeyflowers while I was in my undergrad as well. And what we know is Mimulus lewisii smells like myrcene, so myrrh, like gold, frankincense, and myrrh. It smells resiny. It smells like ossamine, which I think smells like pear drops. And it's got this huge peak of limonene. This is how it's trying to do this attraction, is it wants to smell lemony. Now, the thing that's really interesting, I think, about limonene, it's also why I can't wear perfume, deodorant, or anything scented in the lab, is because you and I smell like limonene. So, the exact same thing that plants use to mediate sexual reproduction, we also use in our courtship behaviors. We are trying to smell like something to make whoever we fancy want to fancy us. So, regardless of whether your perfume is presented as masculine or feminine, 95% of them will smell like this flower. It will be in your deodorant, your moisturizer, and about half of all shampoos and conditioners, right? But I don't really care whether you like the smell of limonene, because I care about pollination, right? And as much as many things are pollinators, including human beings in some cases, giraffes, foxes, all sorts, our focus is on the model systems, especially the ones that we find in Great Britain, like Bombus terrestris. So, how do I know if a bumblebee knows what something smells like? Next question. We have a system for this, which the group leader in our lab, Kelsey Byers, came up with whilst I was at the Gatsby Plant Science Summer School. And this is known as a GC EAG system. This is some real classical science, right? So we take the antennas, we snap them off, it's fine, and we connect them to an electrical system, and as the smells waft over, we see if they elicit a response, because we'll know that that sends a signal through the bee's antenna. So at the top, classic negative control solvent, the solvent that we suspended everything in. Then the green, you got your positive control. This is the pheromone. This is the level of excitement of, oh my God, I am about to get laid. That's what that impulse looks like. And what we know, fascinatingly, and I think really interestingly, is that our flower hijacks that system. And it creates an electrical signal from the same receptors that are producing that response with that lovely lemony smell. So those are the things that we use in the lab, and that's what we think about with the techniques that we're using, and these are the projects we're working on. So first, like I said, is monkeyflowers. They're really like as close as we've gotten to a pollination model, because in a very short amount of evolutionary time, they've made some really, really cool, really specialized systems. So we've got Mimulus cardinalis here, which is here. That is really specialized for hummingbird pollination. The way Lewisii is really specialized for bees. And then Parishii, sulfur. It doesn't want anyone to come, it's actually fine just fertilizing itself. And so what we do is use this as a genetic model. We're doing a lot of crosses, producing a lot of these beautiful flowers like they do here, looking at higher level regulators of scent and scent production as well as pigment. And what we've also done is looked at these species, the Mimulus cardinalis and verbenaceous here, and we recently published some work looking at yellow morphs. So what's really interesting about these really, really red flowers is that when you get to their range edges, right at the edge of where they grow, they will spontaneously fix populations of yellow, which hummingbirds can't see, but bees can. And so what we found was that bees actually respond significantly greater to these yellow flowers due to all of these chemical signals, and also that what we believe is happening is this idea of an incipient pollinator shift. It's the trickling from one pollination mode to another. So the ongoing work that we're working on at the moment with a PhD student in our lab, Eleanor Kennedy, is actually sampling a lot of this Mimulus cardinalis from across North America in this gradient, and we're finding some really cool stuff. So for example, we have a line all the way down at the bottom in Cedrus Island, which decided neither to be red or yellow, but yellow with red stripes. And we're just trying to figure out why that is. The next project that we work on, that I showed you the picture of, is we look at these fragrant floral alpine orchids. We do it because they're so smelly, but we also do it for this reason. In these meadows, you have two parent species, a lovely lilac-y Gymnodenia canopsia that smells like eucalyptus, and then you have Relicani. This is the one that smells like vanilla. And they make hybrids. And in theory, they shouldn't, because it makes a mockery of everything I've just said. Specialized pollination systems, keep yourselves really separate, only have pollinators visit you that will visit other members of your species. Now, they are infrequent, And what we're doing is thinking about what the pollination systems mediate and what isn't being prevented at the early stages, right? Do they actually produce seed? What happens to the hybrids in the second generations? And what we found while we were in the field in 2021 was that they were in full flower, beautiful in August. And we went back 1 week earlier to the exact same individual flowers. We put completely ecologically neutral tags in the ground so we can find the same individuals over and over again. And they were all gone. They had been absolutely ravaged by a really, really, really hot summer. And so what we did was refocus the work. And we have a PhD student called Hannah. She's out there right now. Her entire PhD actually is spending summers in the Alps for the next 3 years. And she's going to be looking at the effect of climate on pollination systems using this model that we've produced in the lab. So that's the research that we do. I did want to do a brief mention of the other things that you can do as a researcher. So one of the things I like to do, and one of the things I think, this is a personal opinion, I think we're bound to do when we take public funding is to tell people about our science. So I feel very fortunate that I've done some science communications and that I've been able to collaborate with some of the organizations here. I am happy to talk about that if that's helpful. And the other thing we need to do is empower the future of science, including every single one of you in this room. But what we know is that there is a deficit of black plant scientists. 6% of the UK identifies as black, but we have 2 black plant science professors in the UK, and you've met one of them. It was Stephen. That's it. And so what we do as part of this collective that I belong to, including Stephen and other researchers, is we're the Black in Plant Sciences organisation. And we run workshops, we fund studentships, if anyone's interested in summer studentships. We also have a meeting happening this October in Leeds, if anyone's near Leeds, or we also have funded bursaries for students if you'd like to attend. And what we like to do is, as it says here, connect, celebrate, and cultivate the next generation of talent. And I think this is really important for thinking about what legacy we want to leave in science going forward. So, with that, I would like to say thank you so much to Gatsby for having me. I'd like to thank Black in Plant Sciences and Reed, which I'm the chair of, and of course the BBSRC pay my wages, and I'm grateful for that. So, thank you very much, and I'm happy to take any questions.